So now let's take a look at the photosystems. This is a photosystem located in the thylakoid membrane, just to give us some reference of what we're looking at. Each photosystem has an antenna complex, and inside that antenna complex is a reaction center. The antenna complex is this blue area here, and it has hundreds of accessory pigment molecules, and this is where the photons are gathered and feed their captured light energy to the reaction center right here. In the reaction center, we'll see one or more chlorophyll A molecules, and they pass electrons out of the photosystem. So in short, a photon of light will come in here and excite one of the accessory chlorophylls in the reaction center. This energy will be passed from one pigment molecule to the next pigment molecule to the next pigment molecule. Like one of those desk toys where you move one ball and it transfers energy all the way through to the next. Eventually it'll end up in the reaction center. At the reaction center, this is where we lose the electrons. The electrons are elevated in energy level, and now they're ready to give us some ATP or NADPH. So again, let's look at the antenna complex. It's also called a light harvesting complex because it captures the photons from sunlight and channels them by bouncing them from accessory chlorophyll to accessory chlorophyll and finally into a reaction center chlorophyll. In the chloroplasts, these light harvesting complexes consist of a web of chlorophyll molecules here shown in green that are linked together and held tightly in the thylakoid membrane by a matrix of protein illustrated by the blue here. Now looking more closely at the reaction center, basically it's a transmembrane protein pigment complex. It consists of three entities. When a chlorophyll in the reaction center absorbs a photon of light, as is shown here, then that electron is excited to a higher level and passed to an electron acceptor, thus reducing the electron acceptor. This oxidizes chlorophyll, leaving it with an electron hole, and then another molecule, a donor molecule, will donate an electron back into that chlorophyll molecule, only for the cycle to repeat again. So light excites the electron, it's raised to a higher energy level, it's passed on to an electron acceptor, thus becoming oxidized, leaving an electron hole, and then a donor molecule will donate an electron to fill that hole. So we have an electron flow, so to speak, through these three entities. So now let's look at the light-dependent reactions. The light-dependent reactions are composed of four essential events. The primary photo event is where the photon of light is captured by the pigment molecule. And then there's a charge separation where this energy is transferred to the reaction center from one pigment molecule to another pigment molecule. And then the excited electron in the reaction center is transferred to an electron acceptor molecule, which is in the electron transport chain. So the third component is electron transport. Just as in cellular respiration, electrons move through electron carriers that are embedded in the thylakoid membrane to eventually reduce NADPH. That's the electron carrier associated with photosynthesis instead of NAD and FADH. These first three events are about the capture of light energy. The fourth event is chemiosmosis. Again, just as in cellular respiration, chemiosmosis produces ATP by transporting hydrogen ions into the thylakoid space. And as that gradient is created, the hydrogen ions want to get out of the thylakoid space, move down their concentration gradient through ATP synthase, exactly the same one that we see in the mitochondria. And in this ATP synthase, as it whizzes through, it spins the motor, which produces ATP. So the first photosystem to be discovered was in bacteria. It generates ATP via electron transport, and it's anoxygenic photosynthesis. So a photon of light will strike one of these accessory pigments. The energy will bounce from one pigment to the next until it finally reaches the reaction center. At the reaction center, the electron is physically removed from the chlorophyll and elevated to a higher excitation level. The electron is then passed to an electron acceptor embedded in the thylakoid membrane. Here, it generates ATP through the process of electron transport and chemiosmosis, just like we see in cellular respiration. So again, this was the first photosystem that was discovered, and thus it's called Photosystem I. Chloroplasts, however, have two photosystems that are connected. This is where we can turn anoxygenic photosynthesis into oxygenic photosynthesis. So the photosystem I is P700. That means that it's most reactive with light wavelengths around 700. This is the one that we see in sulfur bacteria. Photosystem II, however, is most responsive to light wavelengths around 680. And it can generate an oxidation potential that's high enough to oxidize water. We'll look at what that means here in a minute. In chloroplasts, both of these photosystems work together to carry out the non-cyclic transfer of electrons that used to generate both ATP and NADPH. Let's take a step back. When we look here, this is called cyclic phosphorylation because the electron bounces down through the electron chain, generating ATP with chemiosmosis, and returns to the same reaction center that it left. So it's a cycle. However, in conjunction with photosystem II, the electron transfer becomes non-cyclic and we're able to generate NADPH in addition to ATP. In chloroplasts, photosystem II comes before photosystem I. So that is in all plants. Photosystem II comes first and photosystem I comes later. Now, how do we remember which one's P680 and which one's P700? P700 is the higher number and it's photosystem I. P680 is the lower number and it's photosystem II. That's how I keep it straight. So again, electrons are carried in pairs. So let's consider a pair. A pair of chlorophylls in the reaction center of photosystem II will absorb 2 photons of light. And that'll excite 2 electrons, and those will be transferred out of the reaction center to the first electron acceptor, which is plastoquinone. You don't need to know that. The thing to note here is the loss of electrons from the reaction center has now created a hole, an electron hole. So the electrons move down through the electron transport chain to another electron acceptor, plastocyanin. You don't need to know the name of that either. But do remember that as this happens, hydrogen ions are being pumped into the thylakoid space, inside the thylakoid membrane. And then they're going to move through ATP synthase and form some ATP. These electrons in non-cyclic flow will land in the reaction center of photosystem I, because at the same time, photosystem I had a pair of electrons that became excited by a photon or 2 of light. Concurrently, photosystem I is also receiving photons of light, and it works in the same way as photosystem II did. The energy is bounced from chlorophylls to chlorophylls and eventually to the reaction center. The 2 electrons then physically leave the reaction center and are passed to some electron acceptors. Here, NADP is the final electron acceptor. NADP plus binds with 2 hydrogen ions and 2 electrons, just like any of the other vehicles for electron transport, and becomes NADPH. So in this system, we've generated some ATP and we've generated some NADPH. For each 2 electrons excited, we've had 2 electrons excited here, and so we have an electron flow. This electron flow, however, leaves a hole in the reaction center of photosystem II. This electron hole needs to be filled. This is where water is consumed in photosynthesis. H2O is split into 2 hydrogen ions, 2 electrons, and 1/2 of an O2. The 2 electrons are donated into the reaction center. Once this electron hole is filled, both photosystems are once again content. More photons will continue to strike these accessory pigment molecules, which will bounce energy around until we finally excite electrons to leave from the reaction centers. Let's watch a quick animation of the different photosystems to see if it can help our understanding.
In most photosynthetic organisms, thylakoids contain pairs of photosystems called Photosystem I and Photosystem II that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems I and II simultaneously. We will examine what happens with the photons striking photosystem II first. The energized electrons are passed from the reaction center of photosystem II to an electron transport chain. The electrons lost by photosystem II are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem II are shuttled to photosystem I. Within photosystem I, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma.
So to review, first of all, photosystem II is having excited electrons pass to the electron transport chain for the formation of ATP. Those electrons are then deposited in photosystem II after photons of light have excited electrons from photosystem I. The electrons from photosystem I are passed into another electron transport chain, and they are received by NADP reductase, which reduces NADP+ to produce NADPH. The bottom line here is that we have produced, for each pair of electrons out of photosystem II, we've produced 1 ATP. And for each pair of electrons that left photosystem I, we have produced 1 molecule of NADPH, carrying 2 electrons. But as we'll see, it takes more than a one-to-one relationship between ATP and NADPH to fix carbon in the light-independent reactions. In fact, it takes 1.5 ATP per NADPH, or 3 ATP for every 2 NADPH. This diagram below shows you this relationship. 1 ATP is produced from photosystem II, 1 NADPH is produced from photosystem I. In order to get the extra ATP necessary, photosystem I can shortcut and pass its electrons into the electron transport chain of photosystem II. So basically, it short circuits to make an extra ATP via cyclic phosphorylation. So in plants, it takes both non-cyclic phosphorylation, where we do this Z scheme. Photosystem II supplies electrons to photosystem I, which then supplies electrons for NADPH. Electrons have left the system and water has been split to fill the hole in photosystem I. However, every other time it's necessary for photosystem I to short circuit and pass its electrons into the electron transport chain that produces ATP, rather than the electron transport chain that produces NADPH. So in order to get the 3-to-1 ratio necessary, the Z scheme will run twice to produce 2 ATP and 2 NADPH, and then cyclic phosphorylation will happen one time in order to produce an additional ATP. This is another schematic to represent the photosystem II and photosystem I. I like this representation because it gives a nice understanding of what's happening. Basically, a photon hits here, excites electrons, and they're passed to the guy at the top of the tower. He rolls them down through the ATP mill, and we produce ATP. Once the electrons land, another guy puts his photon hammer on there, excites the electrons to the top of the tower, and drops them in the NADPH bucket. So here in the figure below, we've summarized all 4 steps of the light reactions in order to produce ATP and NADPH. Just as in cellular respiration, we have a hydrogen ion gradient created, where the hydrogen ions are going to run through ATP synthase in order to form ATP. Electrons that leave photosystem I will be dumped in the NADP plus bucket to form NADPH. Both the ATP and the NADPH will be carried to fuel the light-independent reactions that we call the Calvin cycle, outside the thylakoid and in the stroma of the chloroplasts. So let's take a try at diagramming the light reactions. What I've begun with here is photosystem II and photosystem I. You'll recall photosystem II is P680 and photosystem I is P700. I'll begin with a photon of light hitting P680. As this photon of light hits P680, it'll excite electrons, and the electrons will pass their energy around to different chlorophyll molecules until eventually they reach the reaction center. Electrons in the reaction center will be elevated in energy level. 2 photons will give us 2 sets of electrons. At the same time, photons of energy have hit P700 in the photosystem I. They bounce around in the antenna complex until the energy is passed to the electrons in the reaction center. Electrons in the reaction center are then shot up to another energy level. Keep in mind, 2 electrons mean 2 photons of light. Electrons from photosystem I will bounce down the electron transport chain and eventually land up filling the hole that was created when photons left P700 photosystem I. In this process of electron transport and chemiosmosis, ADP and inorganic phosphate come together to form an ATP molecule. The electrons from photosystem I have returned almost to the lowest energy level, and they're replacing the electrons that were shot up out of P700. These electrons will bounce down a short electron transport chain, at which point they'll be dumped in an NADPH bucket. So now we have one series of non-cyclic photophosphorylation. We have 1 molecule of ATP produced, and we have 1 molecule of NADPH produced. This scheme will run twice. After it runs twice, we'll have 2 ATP molecules and 2 NADPH molecules. In order for us to get the third ATP, we need one more pair of electrons to produce one more molecule of ATP. So a third set of electrons leave P700. They're elevated to this excited level. And then they can short circuit and dump the electrons into this chain where they go through the ATP mill and produce more ATP. These electrons return to P700, filling in their own hole. By the time we've finished one non-cyclic and cyclic times 1, we get another ATP. With these two together, we'll end up with 3 ATPs and 2 NADPH. Each of these are necessary to fuel the light-independent reactions. We'll see how those come into play here shortly. There's one more piece to the puzzle. There's an electron hole over here in Photosystem I. This electron hole needs to be filled with electrons from a donor. Who was the donor molecule? Well, the donor molecule, you'll recall, was H2O. We split H2O and we send electrons into this electron hole. And we generate some oxygen. This is where oxygen is produced and water is consumed by the light reactions of photosynthesis. So this is a great start to a diagram. See if you can work through this, and I'm sure you'll have some questions. So please don't hesitate to contact me with questions about how to diagram this or anything else in the system. It's really tough stuff to understand. Watch plenty of animations and see if you can sort out all of this information. Okay, to finish off this section, let's take a quick card quiz. What high-energy molecule is the final product of photosynthesis? Where is the chlorophyll located in the plant? A particle of light is called a... What happens at the reaction center of a photosystem? Which of the following is made during the light reactions? Here are your answers. If you got all of these, you're well on your way to understanding what goes on in the light reactions of photosynthesis.